Thank you. So if you look at my name, you'll see that I'm not a Swede. You see this funny letter here. So I'm from Norway. And I'm a professor in medicinal chemistry here at Göteborg University. And we do in part work on things that relates to this topic that I'm going to talk about today. If you're interested to find out more, you can go to the web page, look at it. Now, the idea I had with this was that we're first just going to look at a few things. What is medicinal chemistry? What is this all about and why is it useful? So we're going to spend the first minutes to define a few parameters, a few things that is good to know before we start to look at antibiotics themselves. So on this slide, you will see a definition of what is meant by medicinal chemistry. And if I'm going to summarize that briefly, it basically say medicinal chemistry is about making compounds that have biological activity and then turn these into drugs, meaning that we can treat people with them. That's a little bit different from people that do things called chemical biology and things like that, where the main task maybe be to just get biological activity. But you will see soon that to make them into drugs is a completely different ball game. It's a huge challenge. So for the time being, we just say that medicinal chemistry, that's making bioactive compounds and then hopefully turn them into drug-like molecules that can help people treat diseases. And in this case, we're going to talk about antibiotics. Now, a key step here. If you think, and this is general for all drugs, if you go to the doctor for whatever reason, and the doctor might prescribe some sort of medication, very often you will get a pill to take. And what has to happen then with that pill if it's going to do any good to you? Well, imagine you put it in your mouth. What happened? Well, it goes down to your stomach. And in the stomach, some of that drug might be absorbed. And that's very important because imagine that you got an infection out in your hand and you got a tablet to take. You know, you don't rub the tablet on your hand, you take it internally. And it means that the drug has to be taken up from the stomach through something called absorption and end up in the bloodstream to circulate. Otherwise, the drug will not work. Some of it might continue further down and goes to what we call the intestines. And the smaller part here, the smaller intestine, is also where you have some uptake of drugs. Again, it goes into the bloodstream. So then we have learned already that in order for a drug to work, it has to be absorbed. Now, if it's going to circulate in the bloodstream, if you think about it, what is blood made of? Well, it's mainly water. That means that the drug has to be water soluble. If it's not soluble in water, it won't circulate. Then it cannot be transported to your hand where you have the infection. So solubility is important in order to get distribution, to get it out in the body. All your blood is pumped to your liver. And the liver, you can think about the cleaning station. Basically, that's where the body gets rid of all stuff it doesn't like. And drug-like molecules or drugs in general, the body doesn't recognize that as something that is useful for it, so it would like to get rid of it. And in the liver, to get rid of stuff like that, something happens that we call metabolism. And a very easy picture of that is the liver put what we call functional groups onto the molecule to make it even more water soluble. And if it becomes even more water soluble, we can pee it out. So most of the drugs we eat or take, the metabolism of that end up in the pee and we just urinate it out. But this means that if a drug is going to work, A, it has to be absorbed from the intestines, the stomach. B, it has to be water soluble so it can be distributed. It has to have a reasonable metabolism, because if it's no metabolism, it will stay in the body for forever, and that's no good. Also, we have to think of metabolism can turn the drug into something that is toxic for us. So we also have to think about that to test the drug properly. And eventually, we have to make sure that it gets out. Again, if it stays too long, we might get side effects from it. So all these things are important parameters to think about when you're going to convert something that is bioactive into a drug. If you just focus on two of these aspects then. First, absorption. Imagine that on the far left side here now, that's inside our intestines or our stomach. And what is written here, that's a lipid bilayer. That's the cell wall that is facing out towards the intestines or the stomach. And that lipid, it says lipids, that's fat. So things that are going to go over a membrane like that need to be lipophilic. If it's too polar like water, it will never cross a membrane like that. And up here is something we call passive diffusion. It just means that the molecules, when they enter here, they can diffuse over that cell membrane, but it only occurs if the molecule is lipophilic enough to do that. Then sometimes there are proteins sitting in the membrane that do what we call facilitate diffusion. It helps to drag the stuff over. And the last example down here, then we have proteins that actively pull things over, but it requires energy. ATP stands for adenosine triphosphate. We don't have to know how it looks, but think about it like this. This is the major energy source in the body. So to bring the drug-like molecules from this side over here requires energy. That's what this ATP means. That's active transport. These two other ones is called passive transport because it happens more or less by itself. But it again illustrates our molecule need to be lipophilic in order to cross this membrane. But then we get to the other problem. Imagine we're in the bloodstream, and I said that to distribute it, it needs to be water soluble. So this funny thing written in the middle here, that's just a sugar molecule. It's called glucose. And glucose have this sort of functionalities written like OH, 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 and OH. Since this has this oxygen that has a great appetite for electrons, it really likes electrons. It likes to make hydrogen bonds with water. And it's, these dotted lines means it's water somehow get around it and make interactions with it. Everything you put into water that is solubilized, this happens. You make bonding with water. If we took away all these OH groups here, all this carbon skeleton, the rest of the molecule, is not capable of doing that. So if you don't have these kind of functionalities, no water solubility. And then we get to this situation here. On one hand, if we have something that's lipophilic, it's not really soluble in the bloodstream because blood is water. But lipophilic compounds cross cell membranes, so they're more permeable. If the drug is hydrophilic, it's really soluble, but then it has problem to go over the cell membrane. And this basically means that we have to have a balance between, it should be in Swedish, lagom, just reasonable lipophilic and reasonable hydrophobic or hydrophilic in order to work as a drug. And this is a huge challenge. This sounds really simple. It's a simple question, but it's a difficult problem to address. Now, another thing to think about that, and now we talk more about antibiotic, or we can still talk about drug targets in general. If you have a starting point, let's say that somebody in a company or at the university has discovered a molecule that has interesting properties, and we say there might be a potential for this to become a drug. Then they want to develop it all the way over here and end up with a product. Now, there are two ways we can think about that. When I say novel targets and known targets, it means the following. Novel targets, this means that we have a molecule that has some interesting properties. But if you give it to a cell, it does something we're interested in, but we don't know how it does it. So we don't know what it binds to. That's what I mean with a novel target. But if I have a known target, I know exactly what this molecule does, what protein it sits on, which process in the cell it interferes with. If I follow the lower part and so I know the target, then I can make derivatives or make variants of this molecule, and I can have a pretty good guess what they will do because they're similar to the molecule I started with. And based on that, I can predict what sort of properties it has, like solubility, lipophilicity, like we just looked at. And this means that it's quite easy to get compounds that go into what we call clinical phase. So all drugs, first, you figure out what it does, you make sure it has the appropriate properties, but the key thing, eventually, you have to test it on some patients. So there are 3 stages of that. First, you ask a few volunteers to test the drug, and you just see what happens. Then you crank it up to have maybe a few thousand people that you test it on, that usually have the disease that you're interested to check out. And eventually, at the end, you do a huge test with maybe as many as 10,000 patients just to see how it looks, because all of us are a little bit different. So one drug that might work very well on Anne might have really bad effects on me. So we have to test in a lot of humans, get statistics on it and see what happens. But the key thing you should remember now is, if I know the target, it's quite easy to get up here, but the compounds might fail when we get to clinical trial for various reasons. We'll look a little bit on that later. If you go up here, we don't know what it does, so we have to figure out what's the target, how it does its job. Then we work on new and novel structures of the compound. We have no idea really how to go ahead. It's a much bigger challenge. We have no idea what sort of toxic effect it might cause when it goes to the liver. It might be the bioavailability, which means does it enter the body? Is it taken up? Might be terrible. We don't know that. But the compounds that get all the way here, they're usually quite interesting because they do something novel. They do something new. So then, again, the take-home message. 2 different targets, or 2 ways to do it. Novel targets, known targets. The last case, then we get quite a few compounds up here, but we might lose them in the clinical trial. And up here, we get a few compounds, but they're usually much more interesting. We might lose them too. Then we get into the medicinal chemistry. Look at this, it's called structure-activity relationship. That's, people like me, we do that all day long. Basically what this means is that the structure of the compound, actually how it looks in 3D, it has an implication on what sort of activity it's going to have. So it means if I change the structure, I might change the activity. And I just added a picture to illustrate it. If you can think that the target where our compound should bind is the glow, when I do this structure-activity relationship, I basically want to find the hand that perfectly fit the glow. I can test one hand that might miss a few fingers, and it fits reasonably well, but it's not perfect. It might be that I don't know that I'm looking for a hand, so I try a foot or a knee or whatever, but it won't fit in, so that's hopeless as a drug. So basically, if this is the target structure, I'm trying to do variants of the hand to see what fit best. That's what structure-activity relationship is all about. And it enables us to determine the chemical groups, the functional group that really is important for doing this. If you just think about this picture now, we're going to look at some molecular structures after a while, and you will see what I mean in more detail. So if we are going to make an antibiotic that is useful, and hopefully a new one, one of the goals is to kill or stop the bacteria from growing. If the bacteria cause an infection in us, we want to get rid of it. So we need an appropriate target to do that. And in order to find the perfect match for the target, we do structure-activity relationship. It has to have what we call appropriate pharmacokinetic properties. It's a horrible word, this, but it basically means what the body does to the drug. You remember on one of the first slides I showed that in the liver, the drugs goes in there and the liver do metabolism. That's a very important part of this, because it's what the body do with the drug. But it also has to have appropriate pharmacodynamic properties. That means what the drug does, what the body does to the drug, what the drug does to the body. That means what do they interact with? Does it have any side effects? That sort of stuff. All this is really important. And then what you're going to end up with is that to actually develop a drug that is useful to treat the disease, it's really like looking for a needle in a haystack. It's really, really challenging and much more difficult than you can imagine. From you start out with a compound to develop it and get a drug out of it, it usually takes like 10 to 15 years. It has a cost in the order of 1 to maybe 7 or 8 billions. So it's a massive investment. And the major cost of all this comes from all the clinical trials you have to do, testing on the patients. And when I tell you this about the costs and how much effort it is, you might understand why universities doesn't do very much of this. We don't have the funding, we don't have the resources to develop drugs. Okay, now you know some basic stuff about medicinal chemistry. So now we're going to look at some antibiotics. And this might look very sort of complicated, but think about it this way. In all the boxes are classes of antibiotics we have today, and they are used for what we call systemic use. You taste the word systemic. Can you guess what that means? Yeah. So it means that it's not if you have a local infection on your skin, you take the drug on it, but you take this as a tablet. It's distributed in your whole body. Systemic use. And you see that there are 1, 2, 3, 4, 5, 6, 7, 8 major classes. Sounds quite a lot, because in each of these there are quite a few drugs. And if you look on the top one, I've shown you some more details. And this one you might test it out yourself. It's pretty common that if you get a bacterial infection, go to the doctor, they give you penicillin. So that's a common one. And then in the same class you find a few other ones, Cephalosporins and carpenaments, and also this one. Those that are shown in this sort of gray color, they are the only antibiotics that are synthetic, meaning they're synthesized from scratch in the lab. All these other boxes that are white or yellow are either pure natural compounds or what we call semi-synthetic compounds. We get them from nature and we modify them slightly by doing some chemistry on it. So it means that the largest source of antibiotics, that's nature, not mankind. The ones shown in yellow here are stuff we use to treat tuberculosis. And maybe you heard about that on this course already, but you know, tuberculosis was believed to be a disease that we got rid of. It shouldn't exist anymore. That's not true now, because you have quite a few cases, especially in Russia, and there is no really treatment for it anymore due to resistance. I've drawn up a few structures here. These 3 first one here, they belong to penicillin, cephalosporins, and carpenaments. And we're going to use them as an example to look at some properties. And the last one here is a quinolone, and that's a completely synthetic analog. So the take-home message of this slide, we have quite a few antibiotic drugs. It's plenty of them. The problem is that not all of them are so efficient anymore, so we need to fill up with new ones. That's the challenge. Again, if you're going to do it very simple now, I try to draw a bacteria here. And in the box here, in the letters, there are some processes that are outlined, and it's basically 4 different ones. So the bacteria has some DNA, and it uses that to replicate itself. And there is some transcription to what you call mRNA that are translated into proteins. And then we can say that one class of antibiotics, they interfere with either replication or transcription. So for this presentation, we're going to call that class 1. The second class, it does something with the translation to the protein. The third one, it does something with the cell wall of the bacteria. And the fourth one, it does something with metabolites. It just means things that are produced in a cell or a bacteria that the bacteria need to live. So the simple picture again, 4 different classes. And now we're going to look at some structures in more detail. So this is a quinolone. And when you see this R group, it means that the R group can be whatever. We can have variations there. So the red structure is what is called the basic structure that defines it as a quinolone. And quinolones are completely synthetic agents, all man-made. They interfere with replication, that's the main mechanism. And down here is written what they do in more detail. They interfere with 2 different enzymes. That's not so important to remember, but they do something with replication. It means that the bacteria can't replicate anymore. Don't get shocked by the next one here, because this might look very complicated. Again, you see the basic structure of this. And I've drawn a ring around these 2 chemical groups. It says it's critical for activity. So it means if I take this molecule and, for instance, remove this functional group, the antibiotic is dead. It won't do anything anymore. That's what I mean by essential group. Now, since I'm a medicinal chemist, if I get a molecule like this and I don't know too much about the target, I will start to do the following. I would say, I wonder in position 5, if I change that to various groups, what will happen with activity? This is structure-activity relationship. And then what they found out by doing this, they say that this group here gives better activity than this group. That's even better activity than this group. So it means the activity of the compound depends on what is sitting in R5, or this position. You move over here, it says that having a fluor atom instead of a hydrogen atom is much better for activity. That's why we have 2 of these signs here. So all these sites, when you modify them, we do something with activity. And if you go into the box on the left side here, you see that if I put on this functional group as R7, and I have an R group here, and the R is just 1 atom, it says gram-negative, activity goes up. It means then the drug works better on gram-negative bacteria. Have you heard about gram-negative and gram-positive bacteria? Yes. Okay, for those who don't know, I'll just fill you in briefly that, it's just a question about 2 types of bacteria which have different cell membranes. One of them has a much thicker cell membrane or more layers, so it's more difficult to get the drug inside and kill the bacteria. So we call them gram-positive and gram-negative. If you have a longer chain here, just an alkyl chain, not just a proton, you see that the activity for gram-positive goes up. So it means, again, having different functional groups, we can probably go for different types of bacteria too. Then I can tell you one thing. Have you heard about something called FASS? Yeah? It's this catalog of all drugs that are approved in Sweden. If you go in and search for quinolones, you will get up this structure, and it says something like the following. When you take this antibiotic, you should not eat things like iron sources and magnesium sources together with it. And if you were a chemist now, you could see that from the structure of the drug. Because if it has these two carbonyls next to each other, it will coordinate to magnesium, iron, etc., and make complexes down in your intestines that prevents it from being taken up to the body. So you can see there is even a molecular reason why you shouldn't add magnesium, iron to your food when you take this drug, just because it has this functional group. That was a little side story. Now, on the previous slide, I showed you some structure-activity relationship, but then I also said on the introduction slide that if the drug is going to work, it has to have other good properties: circulate, distribution, metabolism. And a critical thing when we develop a drug is that we have to make sure it doesn't do bad things for us. If you take this drug and look, for instance, at position 7 now, if I have what is called bulky, it just means something that branches out, that takes a lot of space. If I put a bulky group here, this drug goes all the way into something called CNS, central nervous system. If you have an infection in your hand and take a drug for that, you might not want to have the drug into your brain. But if I make this side chain bulky enough, a side reaction is that it will enter CNS. And that might not be so good. Likewise, also position 7, if I go from a standard alcohol to just this ring without any substituent to this 5-member ring, the gene toxicity increases. That means that these compounds start to interfere with genes we have. That might not be so good either. And if you go the other way, from this one on up to alcohol substitution, it says GABA receptor binding. GABA is an ion channel protein that you really need, and it's important to regulate a lot of processes in your body. And of sure, you don't want the antibiotic to interfere with that either. So then you see that in addition to the structure-activity relationship to figure out how can it bind optimal, we had to figure out what sort of toxic effect does it bring along too if you start to modify it. And on this slide, I just showed a few examples. But in general, to figure out all this, they say that an estimate is that we need to make roughly 10,000 variants of drug and test them all to figure out this. And it's only for the benefit of us as patients. If we don't do it, you can imagine if it's released on the market and suddenly people start fall over and die. No, not so good. If we then just jump ships now we have these other class, which are all called beta-lactam antibiotics. And I should briefly tell you why. You see that all these molecules has a 4-member ring, and in the ring there is this double bond to an oxygen, or O, and a nitrogen. And this here is an amide bond, and when that's sitting in the ring, it's called a lactam. I'm going to tell you a thing that might be difficult to believe, but it's really, really true. Usually we say that compounds, drugs, they should bind to the target and they should be released again. They shouldn't sit on the target all the time, because that means it's difficult to get it out of the body. We just want it to sit it and does what it's supposed to do, and that's it. But all these compounds, they make covalent bonds to a protein. It means that it makes a bond that prevents it from being released again. So it actually destroys the target in a way. And it does that only because it has this 4-member ring. So the antibiotic effect of all these 3 types of drugs is sitting in the 4-member ring. I will show you why in a little while. This might look a little bit crowded too, but what I've drawn here is the general structure of penicillin. probably one of the most known and most used antibiotics we have. And then you see all these functional groups, I put a ring around. There's 4 of them. It says essential. It means if I do one tiny change with it, activity goes down. It won't behave like it should do. The only place I can do modifications is here, the R group. All other things I can't really change very much because then I kill the drug. So that's a very sort of, it doesn't allow us to do a lot of variations. Now, some of this you might have seen before. And I'm just going to tell you, what does these drugs do? The beta-lactam antibiotics. And you see there is a funny mix-up picture here now. It looks like we're on a building site. But if you imagine, if you're going to use concrete, you usually put iron into the concrete. Why do we do that? Make it stronger, yeah. Make it stiffer, more flexible. Or not more flexible, more stiff, basically. If you imagine that the iron we put in here, in the bacteria, we find something that has a similar role in the cell wall. And there's this called peptidoglycan layer. So you have a membrane that is just lipids, but lipids are very sort of floating around, you can move them around. But in the cell wall of the bacteria, you have a peptidoglycan layer, and that functions basically the same as the iron we put in the concrete, stiffen off the wall. So the idea now is that if I can prevent the bacteria from making this layer, what will happen? Well, inside the bacteria, the pressure is higher than the outside. So if I take away the iron, it will make a hole in the bacteria and it will die. That's a simplistic view of it. And this might look a bit complicated now, but when the bacteria is going to make that layer, it uses building blocks that looks like this. This might be a little bit disturbing if you're not a chemist, but okay. The top ones here, these are just letter abbreviations for something called amino acids. All our proteins are made of amino acids. And then this is linked to a sugar unit. If you remember the glucose I had on one of the previous slides, this is a similar sugar. And then there is 2 phosphates with a long tail. That is just a carbon chain, so that's really lipophilic. And that is supposed to stick into the lipid bilayer to anchor the whole molecule, to hold it there. And then if you look at the amino acids written there, I've given them some different colors. The first letter, D and L, it indicates something called stereochemistry. There are 2 types or 2 isomers of these. If this was in a human being, it will be only 1 type, because all our proteins is built up of 1 type of amino acids. But you see this one has a mix, and that's only found in the bacteria or microorganisms. That's why we get specificity with these antibiotics. That's why they don't interfere with the cell wall in our normal cells. Now, this is what happens with it. Imagine then that this illustration here, that's the same as this building block. So this black box or gray box is all the lower part here. And these rings in colors are the same amino acids as I have here. Imagine that when the bacteria is going to make its peptidoglycan layer, an enzyme comes in, it has a hydroxyl function on, and since this is an enzyme catalyzed reactions, it attacks here and kick out the top amino acid. That means that the enzyme now is bonded covalently. Covalently means that it's not a weak interaction, it's actually stuck there. Then there is a neighboring building block, and it has a side chain sticking out like this, and this can act and attack again and kick out the enzyme. that means that I have made a cross bound between this building block and this building block. That's how I stabilize my concrete now. I made the iron, so to say. What happens here and happens here is something called nucleophilic substitution. That's a chemical term. It just means that a nucleophile is an electron-rich species, or an atom in our case, that will react with something that has less electrons. In this case, it's the oxygen here that is the nucleophile, and the carbonyl here is the electrophile, and that's why I prefer to attack here and kick out this. And nucleophilic substitution implies that one group replaces another one. So that's a simple definition of it. Now, look at this. I've just drawn a penicillin derivative here to illustrate it. When this enzyme meets one of these molecules, of course the enzymes can't think, but it's easier to say, it thinks that this molecule is the same as this building block. And therefore it attacks it. And the result is that the structure here opens up, but it means the antibiotic is now covalently attached to the enzyme. If I catch all the enzyme on this form, it means I have no more enzymes left to make the iron in my cell wall. So I'm killing the bacteria that way. But have you thought about that then? If you go to the doctor say, yeah, I think you should eat penicillin. And the doctor says, yeah, do that for 10 days or so. And then you go home and you start eating and you think like, I'm not really keen on taking this drug. And actually I feel much better now, but it's only gone 5 days. So I think I stop now. I think that's a wise thing. If you look at this scheme here, and think a little bit about the chemistry involved, you might understand why that's a very, I almost said, stupid thing to do. The reason is that although this is covalent, means that the enzyme is covalently attached, is physically attached to this. This bond there, you see that, that's called an ester. That falls to bits and pieces over time. So if I give this a few days, this is cleared again. The enzyme is all ready to do the same thing again. So, although you feel much better after 5 days, you should take the penicillin for all 10 days, because then you make sure that all these that are released are again caught by the penicillin, so they stay on this form, so you kill all the bacteria. If you stop too early, the problem is that you might kill most bacteria, but then the whole infection blossom up again. And since you stress the bacteria with the antibiotic, they might become resistant if you're really unlucky. And then you have to have a much tougher medication. So it's again, the molecular answer why you should take the antibiotic for 10 days and not stop earlier on. Okay, the question is why it's an ester. The thing is that it's an OH group that does the attack, so this is the nucleophile. And since it attacked the carbonyl here, it automatically forms an ester. If you look at all other places on the molecule, this is also an electrophilic center, but then you won't get the same effect. So it's the only opportunity the drug have. So it always creates an ester. Sure, if we can come up with a smart ways, we can create a bond here that is not an ester but something much more stable. Yeah, that would be better. Now, penicillin again. You might have heard about that already, that bacteria are quite clever when it comes to defend themselves to antibiotics. And sometimes they start to produce an enzyme that specializes on destroying the penicillin. Here again is a penicillin molecule. This enzyme, the bacteria produce itself, and it opens up the four-member ring. If you go back to the previous slide and say that we need to have it on this form in order to catch the enzyme, it means if the bacteria destroy the antibiotic before it can do its job, it doesn't have any effect anymore. So what can we do to prevent that? Well, if you think about it, this is a small molecule. This is a big, chunky protein. So, you know, if the molecule is my fingernail, the protein is probably much bigger than my fist, and it's supposed to sit down and do a job on this. And it means that it has to have enough space to get close to it. So one trick one came up with was that this R group here, if you make that big enough, it's not possible for the enzyme to reach in here and hydrolyze and open up the four-member ring. So if you put groups like this, just imagine that these are quite bulky. So it just becomes difficult for lactamase to come in there. That's the way to get a better antibiotic. And if you think about this like some sort of first generation penicillins, now we're talking ages ago. This is second or third generation penicillins, and today we have the fourth, fifth, I think even the sixth generation penicillins, where we modify this to prevent these things from happening. But the basic structure is still the same. If you look at that molecule to the far left, can you see that that's slightly different from what I looked at on the previous slides? It still have the four-member ring, but the penicillins that we just looked at, they have a five-member ring here. And now we suddenly have a six-member ring, so it's slightly different. And then we have a tail. And when they look like that, they are another variant called Cephalosporins. They do exactly the same as penicillins, but there is one additional thing, and that is that what is sitting out here facilitates the attack of this one. It means when this attack opens up the ring, the electrons move like these arrows are shown. So when this is a group that likes to leave the molecule, this reaction comes very fast and efficient. So you can say cephalosporins in general are more reactive towards the bacteria than the penicillins. They're just designed slightly differently. But they do exactly the same job, same mechanism. So there, it traps it, and again you see the ester. So again, the whole point is that you have to take your antibiotic the whole time, not stop too early, because the ester will eventually be destroyed. The third kind of molecules you're going to look at that belong to the same class are these ones. And imagine now, if you're at a hospital in specific and you're sick, let's say that you have got a transplantation or something, and it's a risk that you get an infection, and they're giving you the standard antibiotics, and they're afraid that, oh, you got an infection from a bacteria that is resistant. This is usually one of the last classes we try out. That's the sort of last resource thingy we do. If you look at the structure again, it looks very similar to both penicillins and cephalosporins. It's just that we added a double bond here. That means that we have much more strain in the molecule. So this is even more reactive than the cephalosporin. They react even faster than the bacteria. And then up here, they look quite different from both penicillin and cephalosporins. And that is important for how they interact with the target. The point of just showing this is that all three of them are quite like, but you see, again, we have modified them slightly. There are different functional groups on them, and that gives them different properties. So now we're getting back to a structure-activity relationship. Now you looked at all drugs. Now we're going to look at a few new ones. Because, you know, if you read the newspaper, you sometimes can get pretty scared about this. You know, it sounds like we're all going to die pretty soon if you don't come up with some new antibiotics. And to some extent that's true, but it's not as bad as we're going to die tomorrow. But how can we make new antibiotics then that can really help us winning this battle over the bacteria? And here are 5 sort of approaches to do that. I'm just going to say a few words about each of them. One thing is that we can take those antibiotics we have and try to improve them by modifying them, just like I went through now. They went from penicillins to cephalosporins to this more recent type of beta-lactams. That would be one way. We modify them so they get better and try to circumvent the problems with the resistance. We can maybe use old drugs. If you think about this, what it was called the golden age of antibiotics, then we talk about the 40s, 50s, maybe the 60s, when a lot of compounds was discovered, but they weren't developed because we didn't have the appropriate tools, or we had so many, so we just ranked them differently and said, we're just going to develop the best ones. So we could maybe go back and reuse them, or we can use old drugs that are designed for other purposes and see if they might work as antibiotics. There's a lot of companies doing that today. We could test things that we never tested before to see what happens. Nature is fantastic when it comes to producing bioactive molecules. So it's, you can always go out in the garden and you're probably going to find bioactive molecules. Some of them might have antibacterial effect. So that's what we mean by untested new chemical diversity. Since we know much more about targets today, and if we have an exact picture of how a target looks, we can design things from scratch, atom by atom. The pharma industry is very good at doing that. And again, we could rediscover these old antibiotics, which is a little bit like repurpose old drugs. We can go back and see what did we test out before that has some interesting activity and see if we can reuse it. Because today we know much more about how to develop things. I'm just going to show you 3 examples that came out quite recently that might help us to do this. This molecule here is a natural product. It's been used as an antibiotic, and this is also the sort of last resort thing. If everything else fails, we try vancomycin, and if that fails, well, there is not really much else to do than hope that you survive. So this is only used at hospitals. And if you just look at the molecule, you will see that it contains a lot of these oxygens and nitrogens, which means that this is a quite polar compound. And this compound you cannot really take orally because it will not be absorbed in the intestines or in the stomach. So you take it by injection, and it's only given at hospitals. Problem, there are bacteria that are resistant. It doesn't work anymore. So what do we do? For instance, this. If you look at the molecule here, it's all the same except for what's in the red boxes. So I would say it's small modifications. If you add this here, you make the molecule more lipophilic, and that means that it suddenly starts to do other things in addition to what this molecule does. This also does something with the cell wall in the bacteria, but it becomes much better if you add this one. Down here they added a functional group that carry a charge on it, and that makes it more cell permeable, which is a little bit funny because I just told you that we want to have things that are lipophilic, no charges, no polarity. But this really improved things. And here, you see over here we had an oxygen here. Here they've taken away the oxygen and put 2 protons. Suddenly it inhibits the transpeptidase much more efficient. And it said that this molecule, it have several mechanisms of action. This one is believed to have 1. And therefore say, by doing this small modification, we have something that is, I think, in the order of 2,000 times more potent than this, much more efficient. And the risk that we will get resistant is much smaller because it have different kinds of mechanism. This was just published this year. I add the reference for those that are really keen to read this.
Is it possible just to ask?
The question is if this is done in vivo or if it's done by other means. This molecule, the way this was developed in a synthetic lab, there were chemists that started out with this or precursors, molecules that are the starting material for making this, and then they modified chemically. This is a pure natural product. This is what we call a semi-synthetic, because you start out with this and modify it to get to this. So this is pure chemical efforts from a bunch of medicinal chemists. Actually, this molecule was prepared at an academic institution, but it's done in collaboration with a pharmaceutical company. So this does not mean that this is now a drug. It just means that this is a very potent compound that might enter clinical trials and may become a drug one day. This is a second example of a molecule that was reported earlier this year. This is a very good example of that things that has not been tested before. It's a completely new compound. It's also isolated from a microorganism. It inhibits RNA polymerases. Now it talks about inhibiting transcriptions going from DNA to RNA. It's loads of drugs that does that, so that's not unique. But the way this thing does it is quite unique. And this compound has never been reported before either. Again, the interesting thing is that it's highly active against drug-resistant bacteria pathogens. That's what makes it exciting. But again, the compound has been isolated, tested. It was also tested in infection models in animals. So it shows that it really works very well. But it's not been in human beings. It's not been in clinical trials. So God knows how it will go. But it's at least a very good starting point. It shows some very nice properties. And the last one I'm going to show you was a major breakthrough that came in 2015, where some guys isolated this molecule. And it looks very complicated. It's basically a peptide chain, so it looks like a bit of a protein. And then it has a tail with this sort of cyclic moiety on it. And again, the interesting thing about this is that it inhibits the cell wall formation of the bacteria, but it does it in a new way, and it does it by different mechanisms. So then they say again, because it's not only one mechanism, the likeliness that the bacteria will become resistant towards this is low. So it's a useful starting point. The interesting part too with this was that this was isolated from a microorganism that we couldn't really handle before. So think about it this way. We go out in the mountains, the sea, whatever, to find this microorganism. Then we bring them to the lab and try to grow them so they can produce this in large amounts. And the special thing with this was it was isolated from a microorganism that people never succeeded to grow in the lab before. But they developed a new technique to do that. And that's how they discover it. So it means new technique that allows us to study microorganisms might help us to detect these sort of things. Again, this has also been in animal studies, and it shows really promising results. So in some years' time, this might be an antibiotic. The question was, what sort of microorganism this was isolated from? Sorry, I have to ask you to look at the paper, or maybe Anne knows. I'm so terrible with names, so I can't recall the name of it. Now, just to give you something to think about, maybe you've seen these sort of things before. If we had a world without antibiotics that worked, then there were a few things that might become a little bit tricky for us. One thing is that we have, sometimes you go to the doctor and you get antibiotic for things you shouldn't really get it for. But think about it like this. If you go to surgery, transplant surgery, that will almost be impossible to do if you don't have antibiotics. Because it's the risk that the patient will die because of the infections that follow with the transplant is very high. removing your appendix, that will become really dangerous. That's pretty standard today, I would say. There's no risk really associated with it, or at least not a high risk, but this would be deadly if you don't have antibiotics. Pneumonia, that would be a killer again. I mean, today you get to the doctor and say, oh, you have pneumonia, you get some antibiotics, and maybe after a week or so, most of us are quite okay. And especially people that are older, you know, when you reach the age of 70, 80, 90, if you don't have antibiotics, a large or a majority of them will die from this disease. Today it's not considered to be anything serious, but it will if you don't have antibiotics. Gonorrhea, it will be very hard or impossible to treat. It's also a bacterial infection. Tuberculosis, well, that's coming along, but if you don't have any antibiotic to treat that with, you know, it will be a deadly disease again. So a world without antibiotics, it's not nice. That will be difficult for us. So that's why I'm saying new antibiotics, urgently needed. And I hope by showing this now that we've gone through that, you know, medicinal chemist expertise, that's required for optimization of key parameters required for candidate drugs, such as structure-activity relationship, permeability, pharmacokinetics, safety aspects, and formulation. Formulation means what we pack the pills with, what had to be in the pill to get it into your body in the most efficient way. So all these things here are things we have to work with. But we had to discover new antibiotics in order to meet the challenges for the future. Otherwise, this will be our future. Good. Thanks a lot.